Alright guys, so we finished up the structure and function of the eye. One last thing I want to mention before we go into visual pathways in the brain is how exactly this photon of light is converted into an electrical signal in the nervous system. So just kind of following the outline that we had here with this light coming in, going through all the parts of the eye that we already talked about. So I basically just blew this up right here. So we took a part of the retina and we blew this up. And this is basically how it looks like in microscopic form. But so what we have here is, this is kind of the overarching picture of how this photon of light is converted to an electrical signal. And so this is the front of the retina, this is the back of the retina. And interestingly, this photon of light is actually going to go through these cells until it hits these cones right here and these rods. And at that point, there's going to be this G protein signaling cascade that occurs, which is what we're going to talk about here in a second. But yeah, the overarching picture is you're going to have this rod or cone receive the photon of light, and then it's going to then activate these cells right here called the bipolar cells, which also combine with other cells known as the amacrine and the horizontal cells, which will then send information to these ganglion cells. And the ganglion cells will receive the information, and the axons of the ganglion cells are actually what make up the optic nerve. So just right here, this is the optic nerve coming out, and these are all axons of the ganglion cells coming out from that. So one thing to note here is that because there's much less ganglion cells than there are rods and cones, there's going to be a loss of details, especially with the rods. Think about, we mentioned earlier, there's about 120 million rods compared to about only 6 million cones. So we're going to lose a lot of details. But with cones, the reason why cones are are much better for details is because there's usually less cones that synapse with each ganglion, whereas there's much more rods that synapse with the ganglion. And so it's kind of an inverse relationship. The more number of photoreceptors that you have that synapse with one ganglion, the less amount of details you're going to actually preserve. So just a side note there. So again, this is kind of the overarching picture. What's happening? You have the rods and the cones, you have the bipolar cells, and then you have the ganglion cells. So let's go ahead and look at this G protein signaling cascade, which is a very important concept for the MCAT. Okay, I do not want you to memorize this sequence here. Again, do not memorize this sequence here, but I do want you to be familiar with how a G protein cascade works, because this is very, very likely that it could show up on a passage of the MCAT exam where you have either a growth hormone or any other kind of hormone, you have ligands, you have drugs, or in this case, photons of light that activate a certain protein that causes a domino effect or a cascade of events to then happen. So let's just look at this just to kind of give you an idea of what this is about. So again, we'll have a rod here and just blew this up. And so we have these membranes within the rods that contain a protein called rhodopsin. And rhodopsin has two parts to it. It has the opsin, which is actually the membrane protein, and then it has a molecule called retinal. And retinal, when it actually receives the photon of light, is going to be converted from the cis conformation to the trans conformation. And this is going to activate the G protein. So hence, phototransduction, well, the G protein is actually called transducin, which is going to be activated. Right here, I know it's super small, sorry, I'm kind of running out of board space, but there's 3 different parts to the transducin molecule. There's an alpha, beta, and gamma subunit. And what happens is the alpha unit of transducin actually breaks off and activates PDE, or phosphodiesterase. And then PDE is then going to cause the transformation of cyclic GMP to then regular GMP. And with a decrease in cyclic GMP in the cell, this is actually going to cause the sodium channel to close. So if the sodium channel closes, what's going to happen to the polarization of a cell? Is it going to become hyperpolarized or depolarized? What's that? Careful now. If the sodium channel is closed off, so you don't have sodium coming in anymore, so you have less positive charges coming in. Good, it's going to be hyperpolarized. So when sodium is coming in here, the cell overall is more depolarized. But when sodium channel is closed off, it's going to be more hyperpolarized, which is really interesting. And this is the only example that I can think of in our bodies where the activation of the signaling cascade actually causes a hyperpolarization of the cell. And this is how the signal is then propagated. So again, with the lack of cyclic GMP, sodium channels are going to be cut off. And where are we at here? So yeah, this is going to cause the cell to be hyperpolarized, which then in turn will turn the rod off. Okay? And when the rod is off, you have a lack of release, or you have a decrease in the release of glutamate. So the amino acid glutamate. And with that decrease in the release of glutamate, you're actually going to cause the on-center bipolar cells. There's off-center and on-center bipolar cells. You're going to cause the on-center bipolar cells to depolarize. And that's going to cause, so once these on-center bipolar cells depolarize, you're going to then cause the signal to be propagated to the ganglion cells and off onto the brain to be transduced. So again, I just want to go through one example there. It is very common, very likely that it could show up on a passage, maybe even a standalone question, less likely on a standalone question, but more likely on a passage. How, you have a sequence of events happening. If you were to take out one or two or whatever, what would the effect of the cell be, or how would that signal then be affected? Something along those lines. So again, this is the last thing I want to go into before we jump into the visual pathways in the brain. But questions on this? On G proteins, kind of how this photon of light is converted to an electrical signal. You guys good on that? Okay.